That means there is an indication that there is something going on, the correlation happened between these two genes. Thus, to concluding our objective 1, we saw that different level of transcriptional diversity in our RT-PCR and AFLR and AFLD genes could be a good marker to differentiate toxigenic and atoxigenic isolates. In the future, we want to see the investigate ATFC, a transcriptional factor. Actually, it is regulate the H2O2 production, hydrogen peroxide, and this hydrogen peroxide linked to the aflatoxin production. We also want to see in future to do some whole genome analysis of our isolates to know better about our pathogen biology. Moving to our objective 2, we assess the co-infection of seed-borne pathogens in peanut seeds using amplicon sequencing. Here we collected our samples from the seedler and we homogenized our sample, followed by DNA extraction and library preparation. And then we sequenced our sample via next generation sequencing, Novogene, and then we analyze our sample through computational tool. And then after that analyzing, we prepared the cluster heat map by relative abundance, species relative abundance. And here we only focus the top 35 genera based on species and species relative abundance and species annotation. And we observed that along with Aspergillus flavus, other species also present on peanut seeds that might co-infect the Aspergillus flavus infection. And some of our isolates also showed a higher relative abundance of fungal genera. So in this step, we don't know the correlation of those species because we didn't do any correlation study. So my take home from this experiment 2 is the co-infection of Ascomycota and Basidiomycota group fungi are equivalent, and it might influence Aspergillus flavus infection and aflatoxin production. As I said, we didn't do any correlation study. That's why in future we want to do co-occurrence network analysis of microbial community to understand better co-occurrence pattern and how they trigger each other in the sense of aflatoxin production. Finally, I want to end my presentation to acknowledge my supervisor, Dr. Imran Ali, my lab member and the funding agency, USDA, NIFA, and my university, Alcorn State University, for providing all the resources and support throughout my study. Thank you very much. If you have any questions.